welcome to Superior Science's live science show. We hope everyone's doing well and staying safe. So our topic today is biology. So you're going to see some cool demonstrations from Katie, Alexandra, Stas, and Amy, and then you're going to participate in a fun activity with Michaela. So all these activities have to do with biology, which in simple terms is just the study of living things. So our instructors are going to go more into depth with that. So just sit back and enjoy the show. So first off, we have Katie, and she's doing yeast reactions in a bottle.
Hi guys, I'm Katie. So today I'm going to be teaching you about yeast. So yeast is actually a eukaryotic single-celled organism that's part of the fungi kingdom. So yeast can undergo a process called cellular respiration. So this is how organisms break down carbs to provide their cells with energy so then they can undergo their necessary functions. So often this would be done with oxygen. So this is termed aerobic respiration when it's done with oxygen, but yeast can do it with or without oxygen. So that actually makes them facultative anaerobes, which means that they can choose to use this function if they want. And then when yeast undergoes this process without oxygen, this is actually called anaerobic respiration or fermentation. So basically how this works is the yeast will take glucose molecules, which is like their source of energy, and then they'll break this down, and this will be broken down into ATP, which is their energy currency, ethanol, which is an alcohol, and then carbon dioxide, which is a gas. So here I have a couple different bottles that I have here. So this is the first one. So in this one, I just mixed yeast and warm water. And so as you can see, the reaction didn't proceed like super strong. So there's just a little balloon here. And then in this one, I added yeast, warm water, and then some sugar. So this one just has 1 tablespoon of sugar. So as you can see, the balloon is a bit bigger on this one. And then here, this one has 2 tablespoons of sugar. So this balloon is even bigger. And then my final one has 3 tablespoons of sugar. So this is the biggest balloon by far. So the reason this works is because the yeast will break down this sugar and it will create the carbon dioxide. So the carbon dioxide goes up into the balloon and it blows up the balloon. So it's coming off of the reaction here, so you can see all the bubbles, and then it'll blow up the balloon. So if you add more sugar, so that's like what I've done. This one has no sugar and this one has 2 tablespoons. So it provides the yeast with more sugar to break down. So then therefore it undergoes a bigger reaction and then it gets more carbon dioxide. So yeah, that's all I have for today. Thank you.
Okay, awesome. Thanks, Katie. So now we have Alexandra, and she's doing digestion in a bag.
Hi everyone, I'm Alexandra, and today we're going to be doing digestion in a bag. So this is all talking about the human body, and in our body there's all kinds of biological processes going on at any given time. So what we're going to use for this, we're going to simulate digestion in our stomach. So what you'll need is a plastic bag, and then some crackers or some kind of food. I'm using crackers today. And then some kind of pop. I'm using this bottle of pop I had. Okay, so when we eat, we chew our food and we swallow it, and it goes down our esophagus. But at the bottom of our esophagus, there's a valve called a sphincter that allows food to go into the stomach, and it squeezes shut so that the food or the fluid that's going into your stomach doesn't flow back up your esophagus. So our stomach has a very important job, which is to break down the food you eat. So the process of breaking down food, this whole process is called digestion. So what we're going to do is we are going to pretend that this bag is our stomach, and I'm going to draw, we're going to draw a little stomach on it. Here we go. It's a pretty good stomach. Great. So what we're going to do is we're going to pretend like we're eating, and we are going to fill our stomach up a little bit. So I'm going to put some crackers some crackers into our stomach. We'll put a few more. Let's pretend we're hungry. All right. Now, so the stomach has two different ways that it breaks down food. So the first way is through physical action. So this is just like we have muscles in our arms and we have muscles in our legs, there are also muscles in our stomach. So we're gonna pretend like your hands are your stomach muscles, and we're going to use the mechanical digestion and start breaking up the crackers. So we can see the crackers getting smaller into smaller and smaller pieces. So this is one kind of digestion. Now we're going to skip to the next kind. So we also have chemical digestion happening. So in addition to physically churning the food in your stomach, our stomach uses chemicals in the form of acids to break down food. So more specifically, our stomach uses hydrochloric acid to break down the particles of food in our stomach. So the stomach, and we're gonna pretend that the acid, because pop has enough acid in it, we're gonna pour some pop into our stomach. Okay. Now, it's very important if you're doing this at home, make sure you seal the bag because you wouldn't want this going all over the place. So now we have this nice mix of stuff. So now what we're going to do is keep churning it and let the pop do its work. We have chemical and mechanical digestion happening in our stomachs. So keep churning it. And you can see that even with that little bit of acid that's in the pop, which is sort of like the acid in our stomach, we have a really blended mix in our stomach. So by the time the food is all churned up and ready to leave the stomach, it's been processed into a thick liquid. This is thin, but usually it's a thick liquid that we call chyme. And so there's another sphincter or valve at the other side of our stomach called the pylorus, and that keeps the chyme in the stomach until it reaches the right consistency to pass into our intestines and continue along the digestive tract. So as you can see, with not a lot of work, we have quite the interesting consistency of product in our stomach. So give this a try at home. And it's interesting to think about all the different processes going on in our body at once. And thanks for watching.
Okay, perfect. Thanks, Alexandra. So now we have Stas, and he's doing saltwater density experiment.
All right, ciao guys. So I'm gonna make sure you can see this. Okay, so you're just probably sitting there wondering at home, going, oh, well, I learned about digestion and VAG and about yeast and stuff. And then you're going, Stas, what does salt and water have to do with biology? Well, I'm gonna answer that question for you right now. Salt is super important to all life. So people who are like crazy health nuts and they go, oh, I don't put any salt on my food. You need to put salt on your food, okay? Or at least somewhere. Humans actually do need salt. If you're deficient in salt, you have a condition called hyponatremia. And if your salt levels are below 120 millimolars per milliliter, I think, it's severe hyponatremia and your muscles will start not being able to contract. So you have no control of your muscles and people can actually get very seriously ill from that. So you do need some salt, not crazy, obviously, but you know, the reason you have salt sensors on your tongue is because your body knows exactly how much salt it needs. Like when something's really, really salty and you need to drink water, your body's telling you to just flush out some of that excess salt. So salt is also found everywhere around us. So 96% of all of the world's water is indeed salt water, not fresh water. Now, I know it's not digestion in a bag or anything, but if you really want to see how your digestion works, you can drink that nice mixture that Alexandra had for you in the previous activity. So here, all I have is water, and then water with I don't know if you know what copious means if you're young, but it has a ridiculous amount of salt in there. So like I put a third of this container of salt in that. So here's one guitar pick not known to be great for floating. We're going to drop that. See, right to the bottom, right to the bottom. If you can see that, I apologize. And if you can't, I also apologize. There she is, right there. Cheers. If that doesn't work, just comment, I blame Michael Cochimiglio. It's fine. Then right there, oh, on top, and then that also. Okay, great. Well, it's been lovely teaching you about salt. Salt your food. Don't get hyponatremia. That's all I have.
Okay, awesome. Thanks, Stas. So now Amy's not here with us today, but she made a video for us to watch. So Michael's just going to play that on your screen.
You guys could see that, obviously. Yeah. Okay, perfect.
Hi everyone, so for today's biology demonstration, we're going to explore how water travels through a plant by conducting an experiment with celery. So what I've done here is I've got 2 jars, one with red food coloring and one with blue food coloring. And I've just put some celery in these jars and we're going to leave them for a couple hours and then I'm going to check back and see if anything happens to our celery. So before I cut to me in a couple hours, maybe just think, do you have any predictions about what's going to happen to our celery? Are they going to change color? Are they going to stay the same? Let's find out. All right, so it's been about probably 5 hours and quite impressed with our celery. So let's take a bit of a closer look, but right away we can already see they have started to change color. So just looking right away, we can already see a distinctive color difference between the two. And if we look a little bit closer, we can actually see the leaves and the stem have adopted this blue color of our water. Now this one's a little bit harder to tell, and at first glance it might just look like it's just wilted, but I think it's actually just the color of the water that we chose. But if you look at the very tips of the leaves, it's actually also stained red. And so now we're going to explore why this has occurred. So in further inspection, we can look at the bottom of our celery, and if you look at these little dark circles, this is actually our vascular bundles of the plant, which have turned blue, and that's because they have absorbed the water. And so the water has traveled from our vascular bundles up through our stem all the way until our leaves. So our celery is displaying the process of osmosis, which is the movement of water from a higher concentration to a lower concentration. And in this instance, the higher concentration is our jar of water, and the lower concentration is our celery. So the water has traveled from the area of high concentration to the lower concentration of the celery. Now, if we were to try this experiment again without water and just food coloring, we might not receive the same results. But you can try this at home with different types of food coloring and different concentrations of water and record your results and see if the same process occurs. I hope you guys had fun with this little celery segment and enjoy the rest of the show. Bye!
Okay, perfect. And lastly, we have Michaela, and she's going to be doing an experiment with the strawberry and DNA.
All right, hi guys, I'm Michaela, and for our activity today, we're going to extract strawberry DNA. So first, let's figure out what DNA is. DNA stands for deoxyribonucleic acid. I know it's quite a mouthful, but it's the material that carries all the information around about how a living thing will look and function. So DNA in humans carries information about the color of our eyes and how our lungs will work. Each piece of information is carried on a separate section called a gene. So in every cell, the DNA structures are called chromosomes. Both the DNA and the chromosomes are really, really tiny. So scientists need to use very powerful microscopes in order to see them. And DNA helps cells make a substance called protein, which you may have heard of before because we need to eat protein for our muscles to grow big. The cells need these proteins in order to live. Okay, so DNA is a structure called a double helix. So there's two sides of the ladder connected by little rungs, and then it twists to form a double helix structure. All right, let's get on to the experiment. So I have a bottle of isopropyl alcohol sitting here on some ice to stay nice and cold. You can leave it in the freezer while you start the experiment. And then we're going to create our mixture here that's going to extract our DNA. So I'm going to pull you down so you can see. So in here, I have about 6 tablespoons of water, and I'm going to put 1 teaspoon of dish soap in here. And then I'm going to add about 1/4 teaspoon of salt. And then we're going to mix that together until all of the salt is dissolved. So you don't want to mix it too fast, otherwise you're going to get lots of bubbles. But just until you see all the salt go away. All right, that looks pretty good. We're just going to put this to the side. Now I have a Ziploc bag, and I'm going to put a whole strawberry in there with the stem and all. And we're going to let it sit in the bag, and we're going to pour in our extraction mixture over the strawberry. So carefully pour it into the bag. And then seal it up, trying to get rid of all the air. Okay, now it's time for the fun part. We're going to squish up our strawberry until there's no more big pieces left. So this is going to take a second. So during this time, I'm going to tell you about what's going on in here. So each component of the extraction mixture plays a part. The soap helps to dissolve the cell membranes. Salt is added to release the DNA strands by breaking up the protein chains that hold the nucleic acids together. And finally, the DNA is not soluble in isopropyl alcohol, which we're going to add in later. So that's why we can see them. So I'm just going to keep squishing them up until I feel no more big chunks. The more you squish, the better, but we're going to take out the big chunks anyways in our next step. So now I'm going to grab a big bowl here and a strainer, and we're going to put the strainer on top so that we can filter out all the big chunks that got left behind. So opening up one side of your plastic baggie and pouring it all over your strainer. Now taking your measuring spoon, you're just going to try and squish out the rest of the liquid that might be stuck in your strawberry. Once that's done, you can put it off to the side. And you have your mixture, which we're eventually going to be able to see our DNA. So we're going to pour it into a smaller glass so it's easier to see. And then it's time for the isopropyl alcohol, which is the fun part. So we're going to take it off the ice, and we're going to measure out about a teaspoon of it. And we're going to pour it on the top. Once you pour it in, you're going to want to hold your mixture at eye level so you can watch the DNA strands separate from the mixture. It just takes a second, so we're going to have to wait a little bit. And then I think I'll be able to show you. So if you look really closely here, I don't know if you can see, you can kind of see the little strands forming that weren't there before. Those are DNA strands in the strawberry. And this is visible from the strawberry DNA because they're octoploid, which means that they come in groups of 8, whereas human DNA are diploid, so they're in pairs of 2. And the longer you leave it, the more DNA you'll see. That's all I have.
Okay, perfect. So that's everything today that we have. So thank you guys for watching. We hope you guys enjoyed and that you learned something. And we're going to be posting this video on Facebook and YouTube tonight. And don't forget to like our Facebook and subscribe to our YouTube. We hope to see everyone on Thursday at 2 p.m. Thanks.